Okay, so first of all, I would like to thank the organizers for organizing this such a great meeting, and it's my great pleasure to show you some of our recent works on visualizing translation dynamics and landscapes inside bacterial cells with cryo-electron tomography and sub-tomo analysis. So in the first slides, I would like to remind the translation carried out by the ribosomes, which is one of the most fundamental process inside all living cells. And since the first discovery of ribosomes on the electron microscope, it has been subjected to extensive structural investigations. So many ribosome intermediate structures has been determined, especially for the major elongation phase, which are assembled by a few intermediate states that to carry out the decoding, peptide transfer, and translocation process to add one amino acid to the nascent peptide chain. However, most of the ribosome or translation intermediate structures were determined using in vitro structural approaches such as X-ray crystallography or single particle cryo-EM, and also they are most from model bacterial cells or other model cells. Also, very often antibiotics or mutations or GTP analogs has been used to store the translation in a certain functional state to really determine the structure. So all these factors, they make it far from the native functional state. And also to determine these structures, they need to break down the cells. That means a loss of the spatial organization, which for the translation case, they need to be well organized spatially and temporally inside all cells. Okay, so to tackle these questions, we really want to use cryo-ET and to do in situ or in cell structural investigation of the translation process inside cells. And to start, we select actually one of the simplest bacterial cells called Mycoplasma pneumonia. It is also a human. Oh, I don't know what happened.
Quite a nice landscape.
Okay, so we used this simple bacterial cell as a model system, and this cell, it is one of the simplest bacterial cells, so it's very small, and which means that it can be directly imaged under a cryo-electron microscope, and also collect quite good cryo-ET data. As I show you here, it's a 3D reconstruction, what we call tomogram of a mycoplasma cell. And also, because we don't need to do Philips scanning, and this enabled us to collect quite large cryo-ET data sets of hundreds of high-quality tomograms. And also use template matching. I managed to pick about 100,000 ribosome sub-tomograms or 3D particles from it. Also, we work together with Dimitri Tukno from the Petrochem lab to establish and validate a new image processing workflow that can sit warp, relan, and M. So it really unifies all the image processing steps. Okay, so with the large data and also the new pipeline, we are able to determine the in-cell ribosome structures at 3.5 angstrom resolution inside native cells. And in this structure, the ribosome base pairs and the ribosome protein side chains can be well determined. This also enabled us to actually build the first full atomic model from de novo of these ribosomes from these genome-reduced bacterial mycoplasmas. And in this model, it enabled us to determine actually new structures that has never been determined before. So that is these ribosome protein extensions. And we also saw that these extensions actually wide exist throughout all bacteria. So this is the first time we really use in-cell maps to first show it and determine it. Okay, so now that we have a large ribosome data set from the cellular tomograms, and this gives us actually another unique chance to really determine the dynamic translation process inside bacterial cells. And to do that, I performed extensive computational classification of these ribosome particles using Liang. So after extensive classification, I managed to determine about 15 different ribosome intermediate structures, and the majority of these are within the translation elongation phase. Also most of these are also quite high resolution, between 4 to 10 resolutions. And this movie I saw is actually to try to place these elongating ribosomes in a sequential row, and really you can see the continuous movement of elongating factors and also TRAs through the APE sites. And also, together with these dynamics of factors, we also see the resting and also rotations of the small ribosome simulate. And at this resolution, it is possible to derive pseudo-atomic models. And here also is in the major class, as you can see, for the most important mRNAs and also A and P side TRAs and also the nascent peptide chains. Actually, we can directly see this density inside cells. So with this, we are able to build atomic models for these different translation intermediate structures characterized by different elongation factor and the TRA occupancy states. And we are also able to place them along the translation elongation cycle. So it covers the full decoding, peptide to transfer, and EFG-mediated translocation stages. Okay, also along this trajectory, we are able to again determine the continuous movement of elongation factors and TRAs, and also we are able to quantify the rotations of the small spionate body domain and also swiveling of the head domain, and also the opening of one of the mobile L1 stock of ribosomes. Okay, so actually what is more intriguing is actually we are able to determine not only the structure but also the relative abundance or distribution of the different translation elongation states. And this gives us kind of an energetic landscape of translation elongation inside living cells. And next came actually with the real beauty of doing cryo-ET as really we can try to map back the determined structures back into the 3D cellular volume based on the coordinates and rotations we have based on sub-tomogram classification and refinement. And this really gives us a 3D landscape of the translating machinery or translating ribosome communities inside native cells. And with this, we are able to answer some very interesting questions. So specifically for these polysome organizations, so polysomes are the ribosomes. They are translating on the same messenger RNA. They can assemble quite close in space. Totally, we detect about 25% of all 70S ribosomes are forming or engaged in these polysomes. And also we are able to determine two basic polysome assembly patterns That is so-called TT and TB configurations as defined before. Okay, so also for longer polysomes, we are able to determine the different topologies they formed from these loosely pacted, more variable configurations to this very packed, almost from these helix-like configurations. And here the different colors are from the different translation of functional states based on the previous classification. And with this, actually, there is a long-standing question within this polysome research. That is, when ribosomes within the polysome, when they are elongating forward, are their elongating states synchronized or coordinate or not? So very clearly from these images, you can see that on the overall level, there is no synchronization. But when we look at the local level, we found actually there is regional coordination. That means state that requires elongating factor, especially EFG to bind, are actually more tended to be within the following ribosomes. As these states 5 and 6 stated, they requires the EFG binding to proceed. And also we found that this effect is more significant for, especially for tightly packed neighboring ribosomes within the polysome. And to determine the structure basis, again, I performed a classification of the polysome here. I divide them into pairs. So really there's preceding one, and this is the leading one. And for the tightly packed polysomes, I managed to determine this structure. And this structure, it shows that actually one ribosome protein called L9, it actually can change its conformation, and then the C-terminal of this L1 can really extend into the elongating factor binding pocket of the next following ribosome. And in this way, it locally and also temporarily lock it in certain states to prevent it from collision with the leading ribosome. And this actually matches nicely with the mutation experiment done in other studies. They have shown that this L9 is not essential, but its mutation can increase the kind of mistranslation and also cause the polysome to be more compacted. So combining all this information, we proposed a new local coordination mechanisms for polysome elongation that is mediated by this L9 to kind of avoid the harsh collision between elongating ribosomes within polysomes and keep the translation fidelity. Okay, so I've been talking about the cases in native untreated cells. Next, we also wonder what might the translation landscapes energetically or spatially change when they are perturbed. So then I collect data with cells treated with 3 different antibiotics. So 2 ribosome-specific antibiotic, one binding to the last unit and another bind to the small ribosome subunit. Also a 3rd antibiotic that is not a ribosome-specific, but rather it's I polymerase inhibit. And later I will show you how they receive the translation landscapes. Okay, so for the first ribosome specific antibiotic, chloramphenicol, binds to the large ribosome unit and also the peptide transfer center. Actually, we found that upon treatment in cells, most ribosomes are rather stored in this state with A and P site TRAs. Also, for this state, we are able to directly visualize this chloramphenicol density right in the A-site tRNA, and in this it actually blocks the formation of the new peptide bond. So also you can see there is quite a strong density that connects to the P-site tRNA of the nascent peptide. So there the incoming amino acids cannot form a new peptide bond. Okay, so for the second ribosome-specific antibiotic, spectinomission, that binds to the kind of the hinge region between the body and head domain of the small spionate, we found that actually the landscape is fundamentally changed, and most ribosomes are stored in this pre-translocational state with EFG and hybrid AP and PE site tRNA. And also for this state, again, we can, for the first time, clearly visualize this drug directly inside the cell, right in the hinge region between the body domain and the head domain. And this way it actually blocks the dynamics of the head domain of the small spionate, which has been suggested to be important for the EFG mediated translocation. Okay, so next it is the third antibiotic. It is I polymerase specific antibiotic, and actually quite surprisingly, we found on the treatment of this antibiotic, most ribosomes are also stayed in this pre-translocational state, similar to the spectinomission. But the mechanisms is quite different, and this is because of the transcription-translation coupling. That means the POM, or this antibiotic stored I polymerase, actually serves as a physical barrier to block the relative MRI movement of within the co-elite ribosomes. And in this way, it again stops it in this state right before the MRI translocation. Okay, so combining all these native untreated and antibiotic-treated data, clearly it shows that different antibiotics reshape the translation landscape, possibly with different mechanisms. Also, we are able to actually perform the translation state distribution within each cells we imaged. So within each of the about 600 cells we imaged. So here each column means one tomogram, or one which covers roughly a cell. And we then can actually perform this single cell clustering analysis based on the translation profiles. Quite surprisingly, we found that actually for a certain experiment group, actually there is relatively low variations between different cells. So this suggesting that for a given cell type under certain conditions, the cells might have the ability to maintain relative stable translation landscapes. Of course, again, this can be changed or upon perturbations like antibiotics. Okay, so also we have data to show that these antibiotics, they also fundamentally reshape the polysome organizations, both in terms of the polysome percentages and also the kind of the packaging of these polysomes. So yeah, I think then I next came to my last slide to take a quick summary. I think using cryo-ET and comprehensive sub-tomogram analysis that include averaging, refinement, and spatial organization analysis, we are able to determine the structural dynamics and also landscapes of translation at unprecedented detail inside genome-reduced bacterial cells. And we believe this is the start of exciting time of cryo-ET and also in-cell structural biology with other integrative structural approaches, such as in-cell cross-linking. Okay, and I think the ultimate goal is really, as this nice review highlighted, it really tried to determine the molecular sociology of the cells in atomic detail. And I think the future is bright. And okay, so next I would like to thank a few people that helped me to finish these works. So first, my supervisor Yulia Mohammed, and also many people at EMBL, Martin Beck, Crystal Muller, and many people from their groups, and also our great imaging facility at EMBL. Also, I want to thank our collaborators, Yuri Rapsilver, and also specifically for this work, Svenje, who helped me a lot with the modeling. And also, I want to thank our collaborator, Dimitri from the Patrick Lab, who developed these softwares. Yeah, thank you.